Okay, so my project is on sexual orientation, more specifically the biological differences between individuals who are homosexual or heterosexual. Just an overview on some things that I found. I found some things on fMRIs, neurotransmitters or neurochemicals, genetics, and neuroanatomy. For fMRIs, we know that they measure blood oxygen level dependent, also known as BOLD, by looking at neural activity. A difference in neural communication has been found between straight and gay males, specifically in the thalamus's blood oxygen level dependent activity. In heterosexual or straight men, thalamic EROI blood oxygen level dependent signals moved in a separate direction in the left inferior gyri, and in homosexual or gay men, the blood oxygen level dependent signals moved in a separate direction from the rectal gyri. We know that genetic factors can also play a role in the development of one's sexual orientation. The Genetics of Human Sexual Behavior paper that I found mentions that there are differences in how genetics and epigenetics impact sexuality. More specifically, for epigenetics, we know that they are environmental factors that can affect the expression of different genes. In homosexual women, more specifically those with congenital adrenal hyperplasia, which is exposure to excessive levels of testosterone, have been linked to higher chances of coming out as homosexual compared to the average woman. We also know that having an older male sibling is an epigenetic factor that can increase the chances of becoming a homosexual male. These environmental factors are influencing the way that different genes are expressed that ultimately affect one's sexual orientation. The same article also mentions that genetic factors or traits may differ for women and men in helping to determine homosexuality. In previous classes, we have discussed concordance rates in relation to twin studies. We know that concordance rates are the probability that twins develop a trait given their shared genes. In previous research, we know that identical twins are more likely to come out as homosexual than fraternal twins, even though a specific gene hasn't been identified. To highlight in this picture right here gives the specific concordance rates for these twin studies. In alternative sexual orientation in humans paper that I researched, the authors did mention a X chromosome marker in the XQ28 region that could also be correlated with sexuality. For neuroanatomy, the third interstitial nucleus in the anterior hypothalamus has been found to be smaller in homosexual men compared to heterosexual men. The interstitial nucleus in the anterior hypothalamus is correlated with sexual partner preferences, hence the sizing difference. Over here, the INAH is what the interstitial nucleus is often referred to. Research on other neuroanatomy structures is still in progress, but some research has shown a correlation between sexual orientation and other brain structures. For example, the suprachiasmatic nucleus, or SCN, is about 1.7 times larger in homosexual men compared to heterosexual men. In this specific study, though, the fact that the subjects had AIDS could have contributed to the size of the SCN. There have also been some data that has reported a size difference in the isthmus corpus callosum. It was larger in right-handed homosexual men compared to right-handed heterosexual men. This picture shows the SEN, and this picture shows the isthmus corpus callosum. Dopamine is still being explored in its involvement with sexual orientation, but as we know, dopamine is responsible for a lot of functions like motivation or motor movements, but one of them is its role in attraction to others or the pairing of different couples. A specific COMT gene makes dopamine break down more slowly, which has longer lasting effects on someone and their relationships. This specific dopamine effect may have a strong correlation to the future development of homosexuality in men.